Hello everyone, I'm Richa and I work in the neurogenetics laboratory here in Queen Square, and my colleague Gary McCarthy is also here today. So we carry out the genetic testing for the muscle channelopathy disorders, paralysis, ATS and the myotonic disorders. So there are several ways in which we can analyse your DNA to look for a change that causes a channelopathy. So one of these is DNA sequencing. We look for alterations in all the genes that are associated with channelopathies. And we can also carry out family studies for the particular DNA change that has been identified in your family. So one of the methods of analysis which I'm going to focus on today is whole genome sequencing, or WGS. So the reason this method of analysis is called whole genome sequencing is that we can look at all your genes in your DNA simultaneously. With previous technology, we used to have to look at each gene sequentially, which was quite laborious. So even though this technique looks at all your genes, we can actually focus the analysis on the genes that cause muscle iron channelopathies. And in the neurogenetics lab, we carry out genetic testing for a range of neurological disorders, including Huntington's disease, Parkinson's disease, motor neuron disease, and dementia. So for patients referred for these conditions, the whole genome sequencing analysis focuses on the group of genes which causes these disorders. So essentially, the group of genes which we'll analyse depends on what you have been referred for. So some groups of genes, which we call panels, contain a small number of genes, whereas others will contain several hundred genes, and analysis of these groups of genes will take a lot longer. So the first offer of whole genome sequencing to patients in the NHS was through the 100,000 Genomes Project, which was a large research study, and it's now carried out as a diagnostic test. I'll just go through the process of whole genome sequencing. So you may get referred to a healthcare professional who will find out more about your symptoms and take details of your family history. Then in your consultation with a specialist doctor, if they recommend whole genome sequencing, you'll have the opportunity to discuss the test and ask any questions that you or your family members might have. So the whole genome sequencing test is usually carried out on a sample of your blood, which is sent to the genomic testing laboratory. In our lab, we carry out extraction of the DNA from the blood sample, and the quality of the DNA is checked to see if it's suitable for genomic testing. So batches of the samples are then sent to another lab who carry out the whole genome sequencing laboratory work, and the DNA samples from all NHS genetic labs in England are sent to this central facility in Cambridge. So once that lab work's been done, the data for the group of genes that are associated with muscle disorders is then returned to us in the lab to read the DNA sequence. And we analyse the genes and look for disease-causing alterations in the DNA. So unlike previous DNA sequencing technologies, a great advantage of whole genome sequencing is that a range of different types of DNA change can be detected. And once we have completed interpretation of the genetic change, we issue a report to the referring clinician with the result. So this slide shows an example of a DNA change that's been detected in the DNA of a patient that has led to their muscle channel disorder. So we read the sequence of DNA, which is represented by the letters. So this patient has a gap or a deletion in their DNA of the letters that are between the arrows. So the grey lines at the sides represent the DNA, and the white section in the middle represents the deleted DNA region, or the gap. So with whole genome sequencing, we may get a result which explains your medical condition, and this might affect your treatment and tell family members of their risk of developing problems. We may get a result which possibly could explain your condition, but we need to do further analysis. For example, test family members, or compare your DNA change, like comparing your alteration with other patients who might have a similar condition. As more genes are analysed in whole genome sequencing, a large number of DNA changes are identified, and this might include a gene change which we don't actually understand. We call these gene variants of uncertain clinical significance, and these may possibly have implications for you and family members in the future. And with whole genome sequencing, we may detect a DNA change not related to your condition, and which may also affect the health of you or your family members. And your doctor will discuss the results of these with you in detail. Finally, with regards to future work, if we look at a group of genes in your genome and we do not find a change which explains your disorder, if a new gene which maybe causal is found in the future, it may be possible to go back to your genome data and have a look at that gene. And just to say, your test data and results are stored in a secure database for the NHS Genomic Medicine Service, and this is the part of the NHS that oversees whole genome sequencing. And I just wanted to mention use of the data for research purposes. So patients who undergo whole genome sequencing are asked if they wish to contribute their genomic data to a secure library so that approved researchers may access that data in a form that does not identify them. If you choose to do this, then your data will be helping research scientists to develop medical treatments. Thank you.